what are the challenges? So these are the approaches. So they are the approaches of prospecting new genes and alleles, which are being employed worldwide, and also we are employing them at our center in the project that we have, mega project that we have on bioprospecting of genes and allele mining. Obviously, the challenge is that you say that you have a diverse bioresource, but how diverse they are? No, because they are not characterized actually. Most often, I say that in case of rice, we say that we have 80,000 germplasm models. Mostly uncharacterized. They're all there in gene bank. So what? Let there be. Let they be there. So you don't know how diverse they are, how useful they are. You don't know. How do you isolate genes and identify new genes and alleles unless you characterize them? So this is a real challenge for us. You have been told about phenotyping. Even if they have been characterized, they have been done in the field. For instance, you talk of disease resistance. Unless you have a controlled condition, your phenotyping is all erroneous. And that's the reason why, if your gene cloning and allele mining, those are all failures if your phenotyping is wrong. So even gene mapping, all that you do, they'll be all wrong because your phenotyping is erroneous, because you don't have facilities to do that. So you require also, you have seen, you require a wide array of expertise to handle these issues. You need a geneticist, a plant breeder, a germplasm specialist, a physiologist, a biochemist. You need many different experts, and then it is not really available at one place. And in India, the problem is this trust deficit. The trust deficit is a serious problem in India. And I give my material to you, and you will not even acknowledge what to talk of, sharing the credit with me. So there's a lot of problems. That's the problem. The expertise at different places, and you don't really share. You don't really true sense collaborate, and that's the real challenge. It's a real challenge to succeed, because this kind of programs would succeed if you have people from many different institutions with different background, different expertise coming together and working together, and also taking the responsibility together, and also taking credit together. That doesn't happen. We have the habit of snatching away credit of others. And that kills the whole initiative. That I take credit for all that which has been done, not even acknowledging you and who has contributed so much. In terms of even material, if I get material which you have spent 20 years to generate the material, if I use the material, I should be able to really acknowledge that. That doesn't really happen. It's a real challenge. Not only here in India, even outside, but they're better. They're better than what we are in this country. And germplasms are not really well characterized. As I said, we don't know the diversity pattern. We don't know what exactly the phenotypic expression and what is the extent of expression that takes place. And if you don't have that kind of platforms, particularly drought tolerance, telling it easy. If you have a kind of rain-out shelter, you can characterize the germplasm lines, but it will not be as precise. In the field condition, the kind of dynamics that you have. If you have a cover at the top, obviously that will have temperature influence on that. Microenvironment influence will be there. So it's not really easy to phenotype. And simulating the field condition under artificial situations is not really that easy unless you have that kind of phenomics platforms where you can simulate the field condition to a great extent. Again, not 100%, at least more than 90% if you can do that. And then that kind of facility is not here actually. So you need, these are all challenges. That phenotyping is a real challenge for abiotic stress tolerance, a real challenge. And if you want to succeed and identify the genes which are responsible, the genes which are responsible for tolerance, for complex traits, it's a real challenge. And access to modern tools of genomics, not everywhere, it's not there. Maybe 1 or 2 centers in the country have some access to this. Maybe all the facilities are not yet available. So you have to have access to the tools and techniques of modern tools of techniques of genomics, because they are essential. In today's context, they are essential. And this has been a limitation, and this is essential. And for that, you certainly need sufficient funding, infrastructure, and also support. What are the requirements? So you have to ensure that you have a rich biodiversity. If you don't have variation in the germplasm, so obviously you want to bioprospect. How do you bioprospect? You need a diverse resource to prospect. So that is that you have to ensure. If they are not, you have to really collect from many different diverse sources, not only from the country, even from outside. And very difficult to get germplasm from outside today because of the conventions and all those things. treaties that we have signed, and also the intellectual property protections that has come today in a big way, and the multinationals which are involved in the process. So, and the consortium mode of operation, as I said, because expertise in many different institutions, if people don't come forward and come together, you will not really succeed. You might keep doing for 20 years, you may not succeed, but people who come together, for instance, rice genome, we could succeed because it was an international consortium, and we from India also, there are two different expertise coming together. So joining hands and getting training and exposure and all that. So that is how we could contribute in 4 years, 5 years time. We could finish rice genome sequencing. Otherwise, from 10 countries and expertise from diverse sources, that is how we can succeed. And so also in other tomato genome sequencing, for instance, that paper also has gone to Nature to publish this, etc. So pooling of expertise available in different institutions. That's very, very important, and characterized from them. And in fact, all those challenges have to be met. They are all requirements to succeed actually. If you see that way, if you want to succeed, you have to meet those challenges. If you don't meet, you will not be really succeed. And modern tools and adequate tools. And briefly, I will tell about this, and then I will finish this lecture. So this is a consortium of 36 institutions and 56 crore rupees for 3 years. And that's the kind of dimension of this activity. And there are the objectives. Obviously, objective is to prospect genes and alleles. That's the objective. And we have bioresource. For instance, plant germplasm resource. You have cereals. You can see a wide range of kind of accessions which are available there, millets and grain legumes. So you have a reservoir of genes there, supposed to be, but you don't know because they are not characterized. They are not yet characterized. They are all stored there. Our experience says that the rice germplasm which is there is not as diverse as it is thought to be. That's what we have a mini core collection, 7,000 of them, and we have phenotyped each one of them. And our experience says it is not as diverse as it is thought to be. That's the point actually. We don't really as yet understand our germplasm research. But certainly rice offers advantages because it grows in the hilltops in the northeast to below the sea level in Kerala, and you can see under cold situations in the north, and salinity situation in the Haryana, for instance, and the coastal salinity. So you have a diverse, in fact, tolerance mechanisms available in a single species. See the evolution, beauty of evolution in this particular species. You grow in totally upland situation in the hilltops. There will be rain and not a single drop will be retained there. It will be all flow down the hills, and still rice grows and matures and gives you complete harvest. No loss. And there is also submergence, and after submergence you have regeneration and you have harvest, a good harvest. And then you have also below the sea level. So you have all kinds of diversities in rice itself which is available. Okay. So although the whole germplasm is not so diverse, but rice has diversity, and you have lines representing all the ecosystems. And the evolution has been such that you have variation in all the ecosystems available, for all the ecosystems available. And the microbes, another resource which we are utilizing to prospect genes and alleles. And you can see in deserts, the Rajasthan deserts, where temperature goes up to 50 degrees in the summer, those microbes survive there. In the rhizosphere, otherwise, it's a microbe surviving there. Hot springs, there are some different Manicaran hot springs, for instance. You have hot springs, where in the Manicaran hot springs, people go and cook their food in that. Put your rice there and cook rice, even vegetables. That's the kind of hot spring that we have. And you have microbes growing there. The cellular all mechanisms operating, you have replication operating, you have transcription operating, you have protein synthesis taking place, and everything is happening in that. The cell is dividing, so everything is happening there. So that means there is something which protects all that. So if you can have a gene like this and put it into plants, this will be highly heat tolerant and drought tolerant genotype that you can engineer. There could be a saproponic protein sitting there which can be used to engineer plant species. So you have that kind of microbes and volcanic eruptions. We don't have volcanic eruptions, but there are volcanic eruptions from where the microbes have been prospected and the genes have been prospected. And we know Taq DNA polymerase and vent DNA polymerases, for instance, they are all from volcanic eruptions. Vent DNA polymerase are volcanic eruptions. And they could be source of genes for crop plants. They could also be source of genes for microbes. For instance, you have a rhizobium. You want to release the rhizobium in the field, but they have to survive the field, adversities. For instance, drought, for dehydration, for instance, heat. And if you engineer a rhizobium which is heat tolerant and drought tolerant, that will be having better competitive ability than the other rhizobium. So that you can do for even for microbes. So, and we have a wide diversity and collection now. We have more than 2,000 collections from these diverse extreme environments, and then characterize them and identify the microbes which are tolerant to salt, tolerant to high temperature, tolerant to anoxic situation, and even some of this, that's anoxic situation and all that. So, and collecting genes, and this is another resource which we have included in our work. And you can see in a beautiful resource, but not worked out at all. We don't know anything. What is the mechanism, adaptive mechanism, that sets this species apart? For instance, you have the single hump camel, and that easily survives without much problem at 50 degrees centigrade in Rajasthan desert. In contrast to that, you have one which is double hump, and these are there, very limited of them are there in Nubra Valley of J&K, and this happily stays there at minus 30 degrees centigrade, minus 30 degrees centigrade. See the contrast at 50 degrees centigrade to minus 30 degrees centigrade, and they also stay in, say, 5 degrees centigrade here. See the contrast, how this adaptive mechanism has evolved. What are the genes which are responsible? And what we have done is we have collected tissues and RNAs and constructed subtractive libraries from brain, particularly brain and other tissues, and then identified genes which are showing differential expression of these environments. The same and also different ones. What is happening in these two? So that you understand the adaptive mechanism in this particular species, which is really wonderful species. You can see from the viewpoint of the mechanism. And then another, we have chosen goat diversity. I mean, this country has tremendous diversity for goats, and you can see the Gaddi and the Chegu type of ones, they tolerate 20 degree plus 20 to minus 20 degrees centigrade. The hilly goats. And you can see these ones, the cold region goats. And you can have hot humid region and arid region goats. For instance, Marwari types, they are all in Rajasthan, Marwari desert region goats. And so you have many different types which are available, and it's a wide diversity. And we are trying to understand not only the genetic diversity, but also what is happening at the gene expression level in this kind of goats. If you bring this goat from the cold region and put it in the normal region, what happens? And what is that adaptive changes that take place? Whether it is able to adapt, or if it is not, what is that gene expression problem that kills the goats? So all these alterations we are trying to study to identify ultimately what to do and what is the objective. Objective is to prospect new genes, which would give you not only which will help you understand the process, but also give you opportunities to engineer organisms. Could be microbes, could be plants, even animals which can tolerate adverse environments. Another resource is fish resource. This is another wonderful resource, because you have marine resource, you have freshwater resource, and all kinds of fishes are there. You can, and we are there are 4 species we are taking. This is cold water fish. This is fish which grows under an anoxic situation. No water and under deep down the soil, it remains there and stays alive. This is called catfish. And then these prawns which are there in saline mountains. So we are trying to prospect. First of all, understand, and that is the mode of operation. Sharing responsibilities and also sharing credits. You have for each, for instance, microbe, multiple institutions to address this moisture stress problem. And similarly, rice, moisture stress, a number of institutions you can see. So responsibilities are distributed. So that's how you can facilitate and go faster and achieve the targets of the project. And so also in other species and other traits. And you can see our strategy, and to summarize, the focus is to identify those master regulators, such as transcription factors and signaling components, and which can be directly utilized and immediately utilized, because the number of genes, downstream genes, they regulate, and you can identify them. In fact, a number of them have been identified by now, For instance, drought moisture stress tolerance, you have AVA dependent pathway, AVA independent pathway, and then there could be, there are other pathways. And then you can identify, what are the elements, which they are binding, and if you identify some of them, and then the newer alleles for some of them, and then you can manipulate all those downstream genes, abiotic stress responsive genes. And when you do this, transcriptome profiling and all that, largely you capture these ones. And only some of them you capture. Wherever some of them showing differential expression. And if you can identify these through SNP analysis, associative analysis, then many downstream genes one can modify. And in rice we have identified some of them and manipulating further in transgenic system. Two of those genes, one which is having, for instance, this class. So we have the generated transgenic arabidopsis and validated the function of those, that they are really giving you that kind of phenotype, drought tolerance phenotype. And one of those eighth box category of transcription factors, that has been validated to give you anoxic tolerance, submergence tolerance, which is not known. And newer genes are being discovered. So new genes are being prospected and newer alleles are being prospected for different kinds of abiotic stress. As I was talking about, that to succeed you require bioinformatics support. If you don't have the huge amount of data from transcriptome, from genome, from SNP, and phenotype, all those data need to be put together and analyzed. If you don't have that kind of resource, you will not really succeed in this program. And a small facility we have created, and otherwise also everywhere you will find bioinformatics facilities, but you don't have people to really do analysis. You may have one or two people, but you don't have that kind of training, that kind of human resource is not really available to handle this kind of data. And that is the major problem in this country. In fact, we make softwares for others to utilize, but we don't know how to really help ourselves. And we don't know how to analyze this data, high volume data which is accumulating. And that's the major drawback of this particular aspect, work in this particular area. So we have created a small facility, and where you will develop these people will develop algorithms for analyzing data, because the available softwares, they don't really satisfy all the requirements, and training people and creating facilities and also providing support for data analysis. So we have created a high throughput genotyping facility, spending about 6.5 crore to create genotyping facility, and also we have now about 8 crore which has been spent to create those next generation sequencing platforms. So the Illumina platform, the 454 platform, and those have been also created there in place to support this genotyping. And this is the automated sequencing platform of this is Sanger sequencing for ABI, and also this is the Illumina platform, both PidExpress and then Illumina iScan platform, both of them are there, they're in place. And then you can, as I was telling, that you can go for large scale allele mining using those platforms. And phenotyping facility, we started with a 2 crore facility, and now we have about 20 crore to create that high throughput phenomics facility, where everything will be all image based analysis. So whatever is happening in the water starters, the plant water starters, the soil water starters, maybe Dr. SSI has already told you. So we are creating a facility with spending 20 crores to create that. 800 plants, they can be simultaneously analyzed. They have grown under control condition, one by one actually imaged for all kinds of the fluorescence imaging, the infrared imaging, the near infrared imaging, the hyperspectral imaging, that all imaging platforms are there in one place, and so that they can be all phenotyped very precisely under controlled conditions. What do you gain? What do you gain from this activity? So obviously you gain strength, the expertise, the knowledge, and all that. And besides that, or your funding agencies will say, what is the product? All that. That's the reason why, everywhere the question is, the agriculture universities are irrelevant, the ICR is irrelevant, every institution will be irrelevant if you don't really give product. That's a big question. And if you don't really show that you have products you are commercializing in the form of variety, in terms of technology, in terms of process, you patent, and also in terms of leasing out to the companies. So unless you do that, your existence will be in danger, and that's the big question. And